Hi everyone, I'm Dr. Nathan Babcock, and this is Physical Foundations of Quantum Biology. Before we begin, I'd like to tell you a bit about my own scientific background. I completed my Bachelor in Physics in 2005 at the University of Waterloo in Ontario, Canada, and my PhD at the University of Calgary, Alberta, Canada. Back in those days, I looked like this. Lots changed since then. I went on to do a postdoc in structural biology at Simon Fraser University in Western Canada, and another postdoc studying spin physics at the University of Exeter in southern England. And most recently, I completed a postdoc in quantum biology at Howard University in Washington, D.C. But enough about my history. Let's look at some history of quantum mechanics, in particularly quantum technology. In 1911, fluorescence microscopy was developed, allowing us to see many of the structures inside of cells. By 1933, electron microscopy had been invented, allowing us to see beautiful electron microscope images of the, for example, pollen grains you see in the picture in black and white near the center of the slide. By 1947, semiconductor technology had been developed, which of course we know all in the computers and laptops, cell phones that enable practically every aspect of our technological lives today. And I don't think anyone's unfamiliar with the laser, invented in 1958. By 1977, magnetic resonance had been developed, allowing us to see inside the human body, even inside the brain. And now quantum computing is posed to revolutionize a wide range of areas of computing technologies, possibly including healthcare, where quantum computers will be used to organize healthcare information and perhaps even diagnose disease. If we want to understand quantum mechanics better, then we can look at individual particles of light and matter. Now, all particles also have wave-like qualities, like particles of light. For example, the photon, a single massless particle, shown here as a squiggle line with E equals hf, the equation drawn over it to indicate that the energy of the photon is equivalent to Planck's constant, h, times the frequency of light. Now, this is a key aspect of quantum mechanics, that energy of a photon is proportional to its frequency up to the special constant of quantum mechanics, Planck's constant, and no similar property exists in classical physics. This is important for quantum mechanics because it helps explain the interaction of light with matter, in particular, the interaction of a photon with an electron, a tiny negatively charged particle that's often imagined to be orbiting the atom. The protons and neutrons inside the nucleus are not so critically involved in the interaction of light with matter, but they also are important for understanding the structure of matter, the proton and neutron each being about 1,000 to 2,000 times heavier than an electron. Now, when an electron interacts with a photon, it can be excited. As the photon is absorbed, the ground state is excited to an excited state of the electron. And as the photon is emitted, the excited state decays to a ground state. Now, in addition to all this, we also have an interesting quantum mechanical property known as spin, which is analogous to the idea of a spinning top, except in this case, the spinning top won't stop spinning. The electron is permanently spinning in that sense and has to go around twice just to get back to where it started. So needless to say, quantum mechanical spin is somewhat different than our daily intuitive concept of a spinning top, but nevertheless has the same intrinsic angular momentum that we think of in terms of rotating bodies. Now, there are other properties, other particles in quantum physics, according to the standard model of particle physics. But for the purposes of doing quantum biology, photons, electrons, protons, and neutrons will serve practically all of our purposes. This brings us to the idea of quantum electrodynamics, also known as QED, or the strange theory of light and matter. It describes light emission and absorption by matter, such as fluorescence and phosphorescent effects. It also leads us to the development of something called Fermi's golden rule, a classic equation derived in quantum physics, classic in the sense that it is very well known and a basis for many calculations, such as calculations of electron transfer rates, which are critical to many areas of molecular biology, quantum biology. It also enables the theory of proton and electron tunneling. Quantum electrodynamics is relevant to the radical pair mechanism, which is well known in spin chemistry, and the theory of super and subradiance, which are important effects that exhibit themselves when a set of atoms or molecules are able to collectively absorb or re-emit light. Now, this leads us to an idea of open quantum systems theory, which is a long-standing theory in quantum biology. Unlike conventional quantum theory, which places a quantum system in isolation, open quantum systems theory defines a quantum subsystem in relation to its surrounding environment. Now, this leads to violation of a standard symmetry of quantum mechanics known as Hermitian symmetry. And in some cases, the non-Hermitian formulation of quantum theory has led to discoveries of important new phenomena, even in recent years. And I've provided an example of a good review article you can take a look at and some of the advances in non-Hermitian physics that have taken place recently. Now, I'm not saying that all biological systems exhibit the most exotic non-Hermitian quantum effects, but in general, in quantum biology, because living things are in contact with the environment, in fact, they need to be in contact with their surroundings in order to survive, it's necessary to consider the quantum mechanical systems of biology in the context of open quantum systems theory. Now, we'll come back to this at the end of the talk, but for the beginning, we're going to look at some more foundational aspects. So, in quantum biology, here's our friend, the European robin. He's become kind of a mascot of quantum biology in recent years. But although he's famous for his ability to sense the Earth's magnetic field, actually to see it even, he's not one of the cornerstones of quantum biology, at least not yet. They're very well established. It's very well established that photosynthesis, the ability for plants to absorb energy from the sun, certain aspects of enzyme catalysis, and biological electron transfer mechanisms all involve inherent quantum mechanical effects. But other areas of quantum biology are less well established. For example, the theory of the avian magnetic sense is often based on the molecule cryptochrome, a magnetically sensitive photoreceptor in the bird's eye, but there are still competing models that suggest that magnetic particles, similar to the iron compass needle you find in a normal compass, may actually be involved in the effect. There's been a theory of olfaction, that is smell, based on quantum effects in molecules that remains to be proven, and also some speculation that even protein folding may involve quantum effects to allow the protein to fold from its single-stranded state into the standard shape of a completely folded protein due to certain quantum effects. Now, not all of these effects are proven, and I'm not going to dwell on areas of quantum biology which are not yet thoroughly established. for the purpose of this foundational talk, we're primarily going to talk about, well, let's not get ahead of ourselves. Some of you may be asking, isn't biology just too warm and wet to sustain any quantum effects? Well, that's an interesting question. Because indeed, according to classical physics, the noisy statistical nature of the biological microenvironment can only be overcome in the limit of large numbers, in which physical processes affecting the gross structure and function of the organism are determined by the law of large numbers. So, this suggests that, at least according to classical physics, that we wouldn't see any real structure or function in an organism except on the macroscopic scale, the scale of its gross anatomy, because the microscopic scale is just too noisy. The atoms and molecules are jumbling around, naturally washing out any formation of any structure as it develops. Now, this is a key idea that was considered by Erwin Schrödinger in the 1940s, who emphasized that all physical and chemical laws that are known to play an important part in the life of organisms are of this statistical kind, and any other kind of lawfulness and orderliness is therefore being perpetually disturbed and made inoperative by the unceasing heat motion of the atoms. But of course, this is based on classical assumptions, and we know, in fact, that there is lawfulness and orderliness, even on the microscopic scale in the organism, which tells us immediately, and this emphasis is Schrödinger's, that the classical physicist's expectation, far from being trivial, is wrong. So we know that classical physics cannot predict the kind of effects we see at the microscopic scale in biology. So where is the quantum in biology? Well, biological quantum effects are well established in redox cofactors. These are special molecules found in proteins, in enzymes, that enable the transfer of electrons in order to carry out certain metabolic tasks. And in fact, more generally, in enzyme active sites. These are special molecular sites in the enzymes that carry out chemical reactions. Now, most of these are, in fact, what are called biological chromophores, which is a fancy word for a light-emitting and absorbing molecule, also known as a pigment. Quantum effects have also been explored in allosteric effects. These are cooperative effects in biological macromolecules, where an effect at one place in the molecule would seem to simultaneously affect its function somewhere else. Quantum effects are well known to play a role in van der Waals interactions, including dispersion forces, and they're also increasingly being shown to exist in coherent collective excitations of molecules, such as superradiance that I mentioned earlier. But pigments and proteins are critical for countless areas of metabolic function in biology. Chromophores, these light-sensitive molecules, also known as pigments, absorb light and re-emit light in the form of fluorescence, the process of fluorescence. So these include things like beta-carotene, which makes carrots orange. Indigo is a famous pigment, as well as the royal Tyrian purple, the pigment of kings and queens. Most of you would have heard of saffron, and some of you may have heard of alizarin. Pigment proteins are crucial to enzyme complexes and are almost universally found in active sites of proteins, where they are critical to numerous metabolic tasks. Redox enzymes, the enzymes that carry out electronic transitions, and specifically electron or proton transfers. And here's a graphical image of an electron being transferred from a previous paper of mine with Deland and coworkers. These kind of redox enzymes carry out electron transfer, and frequently, if not always, the redox enzymes involved in transferring electrons contain chromophores, because the same physics that make them good for transferring electrons make them good for exciting electrons and re-emitting light. They have versatile electronic structures, and they often exploit electronic tunneling. They're essential to physiology, and some key tasks as respiration and photosynthesis. So what's so quantum about them? Well, the thing is that these are molecules, and individual molecules are at the quantum limit. That is to say, the active site of an enzyme is considered quantum mechanical because chemical reactions occurring there involve movements and interactions of electrons at such a small scale that classical physics simply cannot describe them accurately. So there is no doubt that individual biological chromophores are quantum mechanical entities that must be described using the principles of quantum mechanics. This now opens up the doorway to understanding quantum biology, and if your eyes haven't started to glaze over yet, well, it's not too late, because we're going to now consider how we model protein active sites. Can we use exact simulations? Not really. It's way too hard. These, even though they seem like small molecules, are incredibly complicated when you look into their electronic structure. So approximate solutions have been developed using Hartree-Fock methods to consider the wave function of a single what's called a Slater determinant, a set of electrons that are properly symmetrized, as they say, according to the necessary quantum mechanical statistics. A density functional theory, which we'll talk about in some depth as we proceed, but it allows us to imagine the electronic wave function in terms of the density of the electron cloud only. Now, if we need to go into a bit more depth, we might use something like perturbation theory. If you've taken an introductory quantum mechanics course or an upper year course in quantum theory, almost certainly you've done some first, second, or even higher order perturbation theory. And this is when it really gets messy, but it allows to actually simulate fairly significant quantum coherent effects. Density functional theory, however, has become a workhorse in molecular biology, and it is in fact the acronym DFT for density functional theory is now by far the most widely used electronic structure method. It has been for some time. In fact, it's used in practically all of chemistry since the 1960s, when Walter Kohn showed that the energy of a quantum mechanical system is uniquely determined by its electron density. This quantity is more easily handled than complicated wave functions in the Schrödinger equation. Kohn also provided a method which made it possible to set up equations whose solutions give the system's electron density and energy. And this method, called density functional theory, has become widely used in chemistry since, because of its simplicity, it can be applied to fairly large molecules. And we see a nitroglycerin molecule to the right here. This quote and the image are taken from the Nobel Prize website, and Kohn and Popel won the Nobel Prize for their work on computational chemistry in 1998. If we consider molecular biology at large, we have a significant biomolecule here from a paper in Catalysts in 2017, where DFT calculations were used on the active site to gain further insight into, in this case, ligand. These are molecular binding kinetics. Now, since the 90s, attempts have been made to apply density functional theory to biological systems, with the understanding that biological systems, as a definition, the term biological systems may be used in reference to a wide class of polyatomic systems, typically organic chemistry. They can be defined as minimal functional units. These are the elements of biochemistry, which perform specific biological functions: enzymatic reactions, transport across membranes, or photosynthesis, as I mentioned before. Now, on the other hand, as this article reminds us, that common sense definition of the term biological systems could just refer to, frankly, any chemical, molecule, or molecular complex which is involved in biological or biochemical processes. So certainly the active site, which is circled and shown as the catalytic site in this diagram, is physiologically significant. It has a structure and function that are crucial to some part of catalysis in this protein system. So there can be no doubt that these molecular structures are truly elements of biology, not just elements of chemistry like individual atoms. This leads us to the question of how we can use density functional theory to understand these systems better. As the most commonly used method for studying quantum systems, we begin with the idea that a quantum system is modeled by its density. We have a set of interacting electrons. These are all quantum mechanical particles. But if we imagine we can kind of ignore the interactions or even the individual electrons, we can just consider them as kind of a cloud, where the density means the same thing as the usual sense when we consider, say, the density of a patch of fog. Now, what's interesting here is that the ground state, the lowest energy state of the electronic system, and all of its properties may be uniquely determined by the density of the electrons alone. Now, this seemingly classical property, like the density of a cloud or the density of mist, can now be used to determine all properties of interest to a physicist or a chemist. And in fact, we can model the whole system if we imagine it to be replaced even by a fictitious system with just one electron. We can imagine the same electron copied over and over again to give us the density of the actual system. And in principle, it would have the same properties. Now, this fictitious system is imagined based on an effective classical potential. And this classical potential, this force field, is constructed in such a way that when the simplified system reproduces the density of the actual interacting electron system, we can now use it to predict all the same effects. So this is an astonishing simplification of chemistry that was proposed by Cohn and Sham in 1965. Now, can we really do this for any interacting system of electrons? Take the ground state, spin density, rho, and have it uniquely determine some effective potential? And this effective potential can now tell us everything about the system. We just imagine a single electron in a force field that acts as if it was a multi-electron system and as if it had all the same properties. Well, this simple assumption does in fact allow us to construct a fictitious system in what's called an effective potential, sometimes called a pseudo-potential, where we have the capital V is a symbol for a potential like the voltage from a battery. VKS is the Cohn-Sham potential. And it is the sum of the external potential, any forces on the electrons from external electric fields or the electric forces from nearby nuclei. We have an effective potential to replicate the Coulomb interaction between the electrons. Of course, even classical charged particles, like charges, repel. And so we need to reproduce the Coulomb repulsion of the electrons as a classical potential as well. Now, both of those are, in fact, intrinsically classical potentials, and this allows us to basically stuff all the quantum mechanics into the last term, Vxc, known as the exchange correlation function. And having buried all our quantum in this term, we now simply ask if there is an effective classical potential that can somehow, almost as if by magic, reproduce all the results of quantum mechanics. It would seem to make quantum mechanics trivial if we can just forget about doing quantum mechanics, find the magic potential that acts as if it were quantum mechanical, even though it is truly a classical effect. Well, it's not so easy. Why does this matter? I mean, quantum physics would appear to be trivial if the exchange correlation potential is a classical functional. That's a function of a function, by the way. However, unfortunately, in general, there is no solution to finding the exchange correlation function. And in fact, quantum computer scientists have proposed that finding the classical effective potential is, in general, just as hard as solving any or all of the hardest problems in quantum computing. Now, this class, known as quantum Merlin-Arthur, is tremendously important for quantum computing applications, and it would seem to suggest that actually doing quantum chemistry is the same as doing quantum computing. Because if we could solve the exchange correlation functional in general, we could then set up our quantum chemistry problem to solve quantum computing problems and do things like break all the encryption on the internet. So it seems unlikely that even though in principle the classical exchange correlation function can reproduce all the consequences of quantum physics, it seems unlikely that in practice we will be able to find a simple solution to this problem. Now, this raises the question, is quantum chemistry, is quantum biology trivial? I know there's an individual who won the Nobel Prize, in fact, last Tuesday, who has claimed that although quantum physics is central to biology, it is trivial. And it is, in fact, in the sense that we can, at least in principle, imagine a classical potential function that reproduces all results of quantum physics. And generally, an effect is trivial if we have a complicated effect that, under some conditions, reduces to a simpler effect. A theory becomes trivial if it reduces to a simpler one, for example, general relativity. Einstein's general theory of relativity, if we remove all the mass, it becomes equivalent to special relativity, a simpler theory. And special relativity for objects traveling at low speeds, speeds much smaller than the speed of light, reduces to Newtonian motion, Newton's three laws. And if you've taken introductory physics and studied kinematics or dynamics, theories of motion, you can, well, even imagine that at zero speed, these theories of motion also become trivial. Motion stop and they reduce to what is known as statics instead. So a quantum field theory, like quantum electrodynamics, is trivial if it reduces to just one particle. This is a general principle in quantum field theory since the 80s, even before that, in fact. And it's interesting that this concept of triviality from quantum field theory applies so well in quantum chemistry, where indeed it is the standard approach to imagine replacing the multi-electron system, removing all the interactions, and assuming it can be replicated by just one particle, one electron. So why this bother? Why can't we just use quantum theory to predict the physical and chemical properties and then use these properties, the results we get, to parameterize our classical models? Sure, maybe we need quantum computers to find these properties, but once we've got them, can't we just use them as parameters in classical approximations? Well, certainly a lot of work has been done in this direction, but unfortunately, and Beardy and I say it much better than I can, the underlying model potentials require sophisticated parameterizations. They generally omit quantum mechanical effects once you've got your parameters, and they lack transferability to other molecule classes. So if you solve the quantum effects for one molecule or one class of molecules, you can't just take your results and use them somewhere else. You have to resolve them all over again. And in fact, as a result, we cannot describe very well any arbitrary chemical reactions or molecular interactions, if at all. Every new problem, you have to redo your quantum mechanics and produce your properties, sometimes even the same problem if some of the circumstances change, require you to solve the quantum mechanics from the start at every step. So really, there's no option to put your quantum mechanics textbook away and just do things classically once you've solved 1 or 2 or 10, so forth, properties of your system. As a result, quantum computing has become one of the major applications for quantum biology. Predicting molecular structure is a key quantum computing application because if we think about a simple biomolecule like caffeine, according to Jeff Welser, the vice president of IBM Research, we think about caffeine. It has about 95 electrons, so it's not really that big. But if you wanted to simulate that exactly on a classical computer, you'd have to have 10 to the power of 48 classical bits. And for reference, there's about 10 to the 50 atoms on the planet Earth. So unless you're planning on commandeering all the atoms in Earth to build your computer, it's not going to work out. And caffeine is a relatively simple molecule. It might not be that biologically important. I certainly enjoy it in my coffee. But if we look at a really crucial molecule like isoalloxazine, well, we can see there's some comparison to be made to caffeine. Isoalloxazine is about twice as big as a molecule of caffeine, but that doesn't mean it's twice as hard to solve. In fact, it's orders of magnitude harder to solve, such that if you wanted to simulate isoalloxazine on a classical computer, you would need more bits than there are atoms in the whole universe. So by comparison, caffeine is actually pretty boring, and of course, it's not as critical to biological processes as isoalloxazine, which is common in all flavin enzymes, which are very important enzymes in metabolism and physiology. So where do we see these exciting isoalloxazines? Well, we see them in riboflavin on your box of cereal, vitamin B2. You see them in a flavin mononucleotide, which is found in various enzymes, and famously in quantum biology, FAD, flavin adenine dinucleotide, which is an important cofactor, the active site in numerous enzymes, photolyases that repair DNA, phototropins that tell plants when to do photosynthesis, cryptochromes, which are important for regulating circadian rhythms and may be important for sensing the Earth's magnetic fields, amine oxidases, dehydrogenases, monooxygenases, and various light sensing proteins all need flavin molecules. Now, these flavins are sensitive to light, both visible and UV light. They're sensitive to magnetic fields. They're sensitive to the surrounding electrostatics. They're sensitive to the presence of oxygen and pH in the environment. They're present to the movement of the surrounding protein. And they're very sensitive to structural modifications if you actually change the structure of the flavin itself. And about 10% of flavins have random structural modifications that you might not expect to see. Now, all of these change the effects in flavin, the properties of its light sensitivity, its redox properties, and so forth. So you can't just simulate flavin in a vacuum or in a certain set of circumstances and expect those simulations to apply in other circumstances. Far from the environment washing out quantum effects, as some skeptics has claimed, the environment determined the nature of the quantum effects in a molecule like flavin. And despite decades of research on understanding flavin, scientists still don't really know how to model the quantum mechanics of flavin in terms of its photochemistry. There's not a real consensus formed yet in terms of what models work best for simulating its photochemistry. And when it comes to its redox chemistry, electron transfer chemistry, that's barely understood at all. So it remains a major open problem in quantum mechanics to understand flavin and characterize it thoroughly. So let's recap. In this talk, we've reviewed the foundations of quantum biology, basic principles of quantum mechanics as they're applied to biomolecules. And we've gone over a few things that may have been surprising if you're watching this video and you've been reading about quantum biology. So let's go over a few things we've learned. One myth that's sometimes circulated in quantum biology is that quantum effects in biology are really trivial, mostly or all trivial. And in fact, we've seen that even these so-called trivial effects may be impossible to simulate on a classical computer. Even though they're trivial in principle, in practice, they're practically impossible to solve. It's sometimes been claimed that biology is too warm and wet for quantum effects. Well, in fact, warm and wet systems are where quantum mechanics was discovered. Brownian motion, the theory of movement of these systems on a molecular scale, was in fact used by Einstein to predict the existence of atoms. And later, Jean-Baptiste Perrin used Einstein theory to actually demonstrate the existence of atoms, for which he won the Nobel Prize in Physics in 1926. So in fact, warm, wet, noisy systems have provided a basis for demonstrating quantum theory practically from the start. Myth 3, sometimes it's proposed that structural biology is not really part of quantum biology, or not generally considered quantum biology. But in fact, biomolecular structure lays the foundation for biology, including quantum biology, because of course, in biology, structure, anatomy, lays the foundation for understanding function, physiology. And so, in fact, all studies in quantum biology begin with the study of the biomolecules involved and their underlying electronic structure. So I thank you for the interest. I hope that this talk has been very enlightening. And if you've enjoyed what you've seen here today, please consider watching out for my forthcoming book, Quantum Biology, Physical Foundations and Dynamical Applications. If you're interested, send me an email, put a pre-order in now, and consider taking the concurrent course. So we'll cover physical foundations, quantum dynamics in biology, and introductory topics in quantum biomedicine in a full 12-week syllabus coming in 2025. Don't miss out. Once again, I hope you've enjoyed this talk and found it very interesting. If you have any questions, don't hesitate to email me, and I look forward to seeing you at a quantum biology conference in the future. Have a good day.